In this video, video 18, we'll describe and explain the significance of the dissociation curves of adult oxyhemoglobin at different carbon dioxide levels, which is known as the Bohr effect, and we'll explain the significance of the different affinities of fetal hemoglobin and adult hemoglobin for oxygen. Now, these two spec points are probably two of the most difficult points on the module spec for my units, so it's not going to be hard, but it will require some concentration and focus, and please make sure you take notes, pause the video and replay if you're not sure of anything. Now, the Bohr effect was first described by a man called Christian Bohr, who was the father of the probably even more famous scientist called Niels Bohr. And Niels Bohr was a man who did a lot of work on physics in terms of the electron model of the atom, and described the probability waves of atoms and electron energy levels, as you probably heard from him quite a lot in chemistry and physics. But Christian Bohr noticed a pattern of oxygen tending to be released from the hemoglobin whenever the oxygen flowed in the blood near tissues that were respiring. But of course, this didn't happen near the lungs. And of course, the lungs, we tend to find that oxygen binds with a high affinity to hemoglobin and doesn't break away from hemoglobin very much at all. So he wondered why there were different patterns in different parts of the body. Why did oxygen kind of know to join the red blood cell at certain parts, but then break off it at other parts of the body? There must be a mechanism behind it. And he found the mechanism by studying what we call the Bohr effect. And for you to really understand how this works, it's a good idea to either read page 64 to 65 in the textbook. Sorry, ignore me. Page 66 and 67 in the textbook, so 66, 67. And understand how hydrogen-carbon ions are formed. Or watch the previous video, video 17, which will describe it for you on the video. But to summarise the key points, if I take some carbon dioxide in a red blood cell, so CO2, and it's added to H2O, the water in the red blood cell, it dissolves and forms carbonic acid, which of course is H2CO3. And I'm sickened to know that actually there is another mistake in the book. If you look at the diagram, figure 1, you will see that it actually says H2CO3-, which is an impossible ion essentially. So ignore that. This is a neutral molecule, it doesn't have a charge. So another example of publishers taking money but not checking their books. Now, it's a weak acid, carbonic acid, and it tends to dissociate into H+ ions, or protons as we also know them in physics, and also HCO3- ions, or hydrogen carbonate ions. And of course we know the hydrogen carbonate ions pass through from the red blood cells to the plasma by diffusion, and therefore the plasma carries the hydrogen carbonate ions for the most part. But we're interested now in the hydrogen ions. And actually we're really interested because that's really one of the reasons why the Bohr effect happens. And what happens is, well, the hydrogen ions tend to compete with oxygen for space. And actually, by binding with a hydrogen ion, the hemoglobin loses 2 pairs of oxygen atoms. So there's an exchange process there. And of course, the amount of hydrogen ions produced is totally dependent on the amount of carbon dioxide in the red blood cell. The more carbon dioxide there is, the more carbonic acid is formed. The more carbonic acid is formed, the more dissociation occurs. The more dissociation occurs, the more hydrogen plus ions are formed. So we can say that the competition, whereby hydrogen is trying to bind with hemoglobin, is caused by an increase in carbon dioxide in the body. And as we said, the more hydrogen there is, the less space there is for the oxygen to bind, and therefore the oxygen dissociates from the hemoglobin and is able to enter tissues for respiration. So let's think of this in terms of the body. So let's draw a muscle. This is a bicep, and someone is lifting weights. Yeah, so there's a lot of contraction of muscle fibers over a short space of time. They're working very hard and they're respiring, and of course they're taking glucose plus oxygen, and they're producing carbon dioxide and water. So within the muscle there's a high amount, or high concentration I should say, of CO2, and of course if there's a high amount of CO2 in the muscle and there's a lower amount of carbon dioxide in the capillaries, the blood capillaries, then of course we know that the carbon dioxide will go from the muscle and it will go into the capillaries by diffusion, and once inside the capillaries it will be absorbed by the three mechanisms. In the blood plasma it will dissolve, it will also be absorbed by hemoglobin directly to form carbaminohemoglobin, but it will also 85% of it will form hydrogen carbonate ions, and of course we just learnt that that will leave some H plus hydrogen ions as well. So this high concentration goes to capillaries and then eventually forms H plus ions, and these H plus ions compete with oxygen, and therefore the oxygen dissociates from the hemoglobin, and now there's a lot of free oxygen in the bloodstream, in the capillaries. So there's an increase of oxygen in the capillaries, and of course there's a low amount of oxygen in the muscles. So oxygen will go from a high concentration to a low concentration and move from the capillaries to the muscle. So that's a pretty straightforward process, and it's driven by the fact that muscles respire, and therefore they're using oxygen, so a low concentration of oxygen, and they're producing carbon dioxide, so there's a high concentration of carbon dioxide, and that process causes dissociation of oxygen, and it's driven by the H plus ions produced by carbon dioxide dissolving in water to form carbonic acid. But of course if I move the diagram up and I show a similar diagram, but this time I draw the lungs. So I'll draw a lung there, and inside the lung you've got the bronchi entering, branching off the bronchioles as we saw before, and of course then you've got the alveoli, which are much smaller than that in scale, but that's just a rubbish diagram. If I zoom into those alveoli, we can see what's going on. I'll move a bit to the left. We can see what's going on here, and actually what happens here is you've got carbon dioxide in the bloodstream carried away from the muscles, so you've got a high concentration of carbon dioxide in the capillaries and a low concentration of carbon dioxide in the alveoli. So of course carbon dioxide goes from, well, any gas really, or any liquid even, goes from a high to a low concentration, down a concentration gradient. So we find that carbon dioxide diffuses from the bloodstream, the capillary, into the alveoli to be exhaled. So there's a lower, eventually once that happens, there's a lower amount of carbon dioxide left over in the capillaries. And therefore, the second thing that happens is that actually in the blood coming to the lungs, there's very low concentration of oxygen because it's been used up by respiration as the blood has flowed around the body. So low concentration of oxygen there, whereas there's a much higher concentration of oxygen as you breathe in air, about 21% of oxygen in the air. So oxygen moves again from a high concentration to a low concentration, so oxygen moves from the alveoli and diffuses into the bloodstream, as we said before. Now, As a result, we notice that there's a decrease gradually as the carbon dioxide diffuses through, a decrease in carbon dioxide, and therefore there's a decrease. I should draw that as a bigger line. So therefore there's a decrease in carbonic acid. If there's a decrease in carbonic acid, there's a decrease in dissociation, and therefore a decrease in H plus and HCO3 minus ions. If there's more oxygen in the bloodstream now, because don't forget that was before, but now the oxygen's diffused from the alveoli into the bloodstream. If there's more oxygen now in the bloodstream, in the capillaries, that means that there's more oxygen, less hydrogen ions, and therefore the competition for the space of the hemoglobin is won by the oxygen. So the oxygen binds and has an affinity to hemoglobin. So that's essentially the Bohr effect in a nutshell. But we tend to draw it graphically, so we tend to use graphs to explain the Bohr effect. But it's better to explain it in words and in diagrams before we get onto the graph. But I'll show you the graph now. And this graph has an x-axis which shows the partial pressure of oxygen per kilopascal. And partial pressure is simply the amount of pressure applied by oxygen as a percentage in kilopascals unit of pressure. And the y-axis shows the percentage of saturation with oxygen, and that's the hemoglobin saturated with oxygen. So of course, ignoring the three lines, just taking any random line, C, B or A, we should expect the natural curve, as you increase the amount of oxygen, or the partial pressure of oxygen, as you increase the amount available, this increases the amount of absorption or saturation of the hemoglobin. So the hemoglobin can hold more and more and more oxygen, the more partial pressure increases. To use a really inaccurate and really crude analogy, again we can use a sponge. It's like saying the more water you apply to a sponge, the more the sponge absorbs water to a certain limit. But of course, once the sponge is completely saturated, it then tails off. But that's not really the point of the graph. The point of the graph is to compare lines A with line B and line C. And that's an extra variable to consider once you've looked at the x and y axis. So it's a third variable. And we see that the graph of A, B and C differ in the partial pressure of carbon dioxide. So A has a low partial pressure of carbon dioxide, B has a medium partial pressure, and C has a high partial pressure. So question, where in the body would you find partial pressures of around C, 10.7 kilopascals? Have a think about it, pause the video and then answer once you unpause it. So you should know that there's an increased amount of carbon dioxide, probably in respiring cells. And the chances are this would be respiring cells in places like your muscles, where there will be a very high rate of respiration during exercise, and therefore a high amount of carbon dioxide being produced and an increased partial pressure. Whereas with A, you probably find that somewhere in, near the lungs, because of course at the lungs the carbon dioxide is diffusing from the capillaries to the alveoli, and therefore gradually decreasing the amount of carbon dioxide available in the capillaries in the bloodstream. And we know why that's true in words. We know that less carbon dioxide means less hydrogen ions produced, which means less competition with oxygen, which means oxygen can bind onto the hemoglobin freely, whereas of course in the muscle tissue, more carbon dioxide means more hydrogen carbonate is formed, which means more hydrogen ions are formed, which means there's more competition for space, and therefore there's less oxygen attaching to the red blood cell. Simple. And the graph is actually just showing you that in numbers. Because as you increase the partial pressure, the graph shifts to the right in this direction, and this is important to remember, and also it shifts downwards. So the oxyhemoglobin dissociation curve, the curve showing how oxygen dissociates actually shifts downwards and to the right if more carbon dioxide is present. So if more carbon dioxide is present, the hemoglobin is less saturated with oxygen, which means the oxygen must be being released from the hemoglobin, and that's exactly what you want. If your muscles are respiring actively and they're producing lots of carbon dioxide, then you're going to want a lot of oxygen to be used for respiration. So it's good that happens, because it means that the hemoglobin doesn't bind strongly to the oxygen is released and it can enter the tissues for respiration. But of course, if you decrease the amount of carbon dioxide, if you go the other way, you see that the dissociation curve, the oxygen dissociation curve, shifts upwards to the left. So we see that A is further up than C and further to the left than C, which means that actually decreasing the amount of carbon dioxide reverses the pattern. And that's good in the lungs, because you don't want your hemoglobin to loosen up and let the oxygen be dissociated. You want your hemoglobin in the lungs, or near the lungs, to bind very strongly to oxygen, because you want the oxygen to be carried around your body. You don't want oxygen diffusing back out of your lungs. And this effect, the Bohr effect, is a completely natural effect, and it means that oxygen binds strongly to hemoglobin. So Albert HBO is strong, whereby the concentration of CO2 is lowered, whereas the hemoglobin and oxygen binding is weak when the concentration of CO2 is increased. And we know why that is, and we know why that's useful, and that is basically the Bohr shift. So you need to explain how the Bohr shift happens in terms of ions dissociation, but you should also be able to describe the pattern on the graph. So we should be able to describe and explain the significance of the dissociation curves of adult oxyhemoglobin at different carbon dioxide levels, the Bohr effect. We'll now cover point M, which is about fetal hemoglobin and oral hemoglobin for oxygen. So like before, if I drew a graph, and on the x-axis I had the partial pressure of oxygen, and it's going to be a really messed up graph because unfortunately it's not working very well. And the y-axis I had the percentage saturation with oxygen. So I'll write this in a second. So that will do. And I've used O2, but I haven't managed to find the subscript button, so there's a big 2 there, so ignore that. And of course, the unit is kilopascals. We also call partial pressure of O2, we can call it oxygen tension. So an increased partial pressure will be an increased oxygen tension, is another word you might find. So if I was to draw a graph of partial pressure of oxygen compared with saturation of oxygen, but I was looking at something simple like water or a fluid, you'd find that it would be literally a direct straight line. So I could draw in another colour and another thinness. I'll use the pencil too. So we'll draw a direct straight line, because the more oxygen that's present, the more dissolves. That's pretty straightforward. However, with hemoglobin, it's a bit different. You see a different shaped curve. You see a sigmoid or an S-shaped curve. So if I was drawing hemoglobin, you would draw actually a curve that looks a bit like this. So we start there, it gradually increases like that, and then it levels off like that. And there's a reason why there's a different pattern in hemoglobin. And the first reason is that if there's not much oxygen available, so if there's a low partial pressure of oxygen or a low oxygen tension, that means there's only a few molecules of oxygen. And hemoglobin is a very big molecule, as you can see. It's got 4 domains, and the heme groups are buried within the molecule quite deeply, and they're in the centre of the hemoglobin molecule. So with a small amount of oxygen in the vicinity, so very small of O2, it's very difficult for a couple of O2 or oxygen molecules to permeate and diffuse through the molecule, and there's simply not enough pressure or tension on the outside to force the molecules to diffuse through. And the rate at which this happens is very slow. Now, of course, if you increase the rate or increase the amount of oxygen outside and change the concentration, the more there's a difference in concentration, the faster molecules will flow down the concentration gradient. And this is why the first part of increase, so the first few times you increase the partial pressure, not very much happens because, as we said, it's difficult for the oxygen to bind. But eventually, if you increase the partial pressure enough, if you increase the oxygen tension around the hemoglobin, the tension soon becomes so much that there's a diffusion gradient. There's a high concentration of oxygen outside the hemoglobin, a very high concentration, and a very low concentration inside the hemoglobin. So this causes the first oxygen molecule to bind with the first heme group. And I'll draw an arrow showing that happening. And once that happens, it changes the bonds in the hemoglobin molecule, and actually the oxygen binding causes a slight change in the shape of the hemoglobin, or the conformational change. So that conformational change changes the structure of the hemoglobin. And this allows a second and a third molecule to quite easily diffuse through the newly structured shape of the molecule of hemoglobin now. And therefore, the graph suddenly shows a huge increase in saturation percentage of hemoglobin as you increase partial pressure. So you've gone from a very slow increase to a very fast increase. But of course, once it gets near the top, it levels off. And the reason for this is simple. Well, there's a few reasons actually. The first reason is there's now a higher concentration of oxygen inside the hemoglobin to kind of almost counteract the concentration outside the hemoglobin, and therefore diffusion is slower. The second reason is because there are already 3 oxygen molecules bound to the 3 heme groups there, it's very unlikely that a 4th oxygen molecule will bind unless it's an extremely high external oxygen partial pressure or oxygen tension. So as you increase the partial pressure of oxygen, the graph starts to level off, and only under very, very, very high oxygen tensions or partial pressures does the 4th oxygen molecule bind onto a hemoglobin molecule. Now, of course, in the lungs, there's an extremely high partial pressure of oxygen because there's a low oxygen content in the blood and a very, very high oxygen content in the alveoli, and therefore we approach almost 100%, and therefore it's possible for the percentage saturation to be as high as possible, which is almost 100%. And of course in the muscles, where there would be a much lower partial pressure of oxygen outside the hemoglobin molecule, that means there's a much lower saturation, and therefore the oxygen can dissociate and enter the muscle cells where respiration can be continued. So that's adult hemoglobin. Let's talk about fetal hemoglobin now. So adult hemoglobin we drew earlier, we showed you this sigmoid or S-shaped curve. Fetal hemoglobin quite simply has the same shape, but it's shifted upwards and to the left. So if I draw this in brown, we can show that the curve actually looks a bit more like this. So I should really level off the same at the top there, so I'll correct that. There we are. Not very accurate. But the point is, we see that adult hemoglobin has the familiar shape of an S. Fetal hemoglobin has a shift to the left and up. And what that means is fetal hemoglobin has a higher affinity to oxygen than adult hemoglobin does. Now it works as follows. If a fetus is growing inside a mother, and I'm going to draw a very crude and badly drawn fetus here, so I'll draw the head, limbs, that'll do. The fetus is growing inside the mother, and therefore the fetal blood and the mother's blood is actually separate. But the mother's blood passes through the placenta, well, not through, but near the placenta, and the capillaries allow exchange to happen. So what happens is the mother's blood contains blood cells that have oxygen bound to them to form oxyhemoglobin, and of course that means the mother's blood has a high partial pressure of oxygen. And this free oxygen in the blood passes through to the baby, or the fetus, sorry, and the fetus can absorb the free oxygen in its blood cell by binding to its hemoglobin. When this happens, this means that the blood fluid, or the plasma in the mother's bloodstream, has a much lower oxygen tension, but the hemoglobin found in the red blood cells in the capillaries has a higher oxygen tension. So oxygen comes from the red blood cells into the plasma, and that means there's even more oxygen for the baby to pick up. So this causes the graph to shift leftwards and upwards, which means the fetus has a higher affinity to oxygen than the mother does, and therefore the mother can supply the baby, or the fetus, sorry, with oxygen for respiration. Now, if that wasn't true, if the baby had a lower affinity, this means that whenever the blood flows from mother to near the baby, or I keep saying baby, I should say fetus, so I'll correct myself again. When the blood flows to the fetus, the mother's hemoglobin would bind more strongly to the oxygen than the fetus's hemoglobin, and therefore the fetus would starve and wouldn't gain the oxygen. So this curve has to show that the fetal hemoglobin is stronger. And you might wonder why the mother doesn't die, and the simple reason is some of the blood is directed towards the fetus, but not all of it. Other capillaries and other arterioles direct blood away from the fetus to the required kidneys and liver and muscles and brain and so on. So of course the mother can still give her own body the oxygen it needs, but the blood that passes near the fetus needs to be given up completely to the fetus. So this was the longest video I've recorded so far, but I do think it's also probably the most difficult. So hopefully you now understand that the Bohr effect is the shift downwards and to the right of oxygen dissociation when there's an increase in carbon dioxide. And this is due to carbon dioxide producing H+ ions, which compete with oxygen for the space of the heme group in hemoglobin. And we should hopefully describe and explain the difference between adult and fetal hemoglobin in terms of different affinities, and we should be able to identify that in the graph and explain why the fetus can gain oxygen to respire.